G'day. Welcome to the AgriCoach Podcast for families on the land who want to grow their family's wealth and prosperity across the generations. I'm your host, Ben Law, and I'm the Financial Bloke. You've probably noticed that we've had a bit of a change on the podcast from the Financial Bloke Podcast to the AgriCoach Podcast, brought to you by the Financial Bloke. It's the exact same host, me, the exact same content, and the exact same objective. It's just a simple name change, nothing more. So now that we've sorted that out, let's get cracking. We're currently going through an unprecedented time in history, with a rate of technological change being at a pace we've never seen before in human history. The speed of change is not only remarkable, it's accelerating at a rate that even the most in-the-know scientists and futurists could scarcely have imagined. Over the past decade or so, 3 monumental shifts have reshaped our landscape. Number 1, the exponential growth in computer chip processing power. Number 2, significant advances towards artificial general intelligence, or AGI. And number 3, rapid changes in the somewhat emerging field of biotechnology. I'm personally fascinated by all 3 areas, and I'm watching them closely with absolute fascination. Now, you're probably wondering, where the bloody hell are you going with this, Ben? Well, last year I had the privilege of having breakfast with Professor Van Eenennaam, one of the world's preeminent researcher in animal technology and genomics. Being the curious creature that I am, I asked Alison this simple question. What are some of the really cool and kind of crazy projects you're working on, or that are catching your mind right now? And what she shared with me was nothing short of mind-blowing. One that stuck with me the most was the research that Alison called switching an animal's balls. Now picture a scenario where a bull sires calves that bear no genetic resemblance to him whatsoever. It's kind of like introducing a Brahman bull into your herd and unexpectedly welcoming Angus calves. It's essentially the genetic material in the Brahman's testicles isn't his own. And now if that doesn't blow your mind, I'm not sure what will. And this was just one example of the fascinating projects underway in the field. So today, we're truly privileged to have Professor Van Eenennaam as one of our guests, all the way from sunny California. Alison is a professor of cooperative extension in the field of animal genomics and biotechnology in the Department of Animal Science at the University of California, Davis. Her research and outreach programs focuses on the use of animal genomics and biotechnology in livestock production systems. So to talk all things cool in animal biotech, without further delay, welcome, Alison.
Thanks, Ben. I appreciate the offer to be here.
Fantastic. It'll be great. I'm really interested in today's topic, so it's going to be a whole lot of fun. Now, to start off with, can you just give us a quick description on what is actual animal biotechnology and what does a researcher in animal biotechnology actually do?
Yeah, fair question. Because really that's a pretty general term, biotechnology. It's life technology, and I would argue, well, that's just farming. And animal biotechnology is animal agriculture, right? You could make that argument, but I think more colloquially it gets used to describe techniques associated with DNA technologies and things like recombinant DNA and GMOs and that type of thing. So I was actually employed in extension in my job to talk to farmers and also the general public around biotech, just because of the kind of the political controversy around GMOs, and because that really created issues for animal agriculture to adopt the technology. So you may be familiar with the recombinant bovine somatotrophin or growth hormone that was genetically engineered protein that dairy farmers could use to increase milk yield, that ran into a lot of activist intervention and misinformation about, giving you cancer or whatever. I can't remember exactly what the scare tactic was back in those days. And it was actually that campaign that led the University of California to actually employ me. So I actually owe my job to kind of misinformation and activists. So thank you, Jeremy Rifkin, and have been really working in that space, not only doing outreach to farmers around these technologies, how to use DNA information in selection decisions, but also research into some of the more molecular aspects like genome editing, which is really a focus of my lab at the moment. So a little bit of a foot in the door of education and outreach and hearing where problems are, and then the other foot in the lab trying to address problems or do some crazy things that I think would be really useful for the sustainability of animal agriculture.
So can we just duck in, Alison? Can you give us an example? Because everyone thinks about as you said, the animal, the activists around, the problems with biotechnology. But what has been one project maybe that's been a real win for agriculture and for the planet around that space?
You can't argue that genetically engineered crops have dramatically reduced the environmental footprint of those crops' production, be it BT or even the herbicide-resistant crops have dramatically reduced the use of more toxic herbicides and what have you. So that's plants, and yep, that's all good, but who eats plants? Well, animals. So in terms of animals, I think perhaps the most successful example I could give you is a little bit of a silly example, but it's an aquarium fish that you can buy over here in the United States that's carrying jellyfish fluorescent genes. So there's fluorescent orange and green and red aquarium fish that have been genetically engineered just for hobbyists, enthusiasts. And I think it's the most successful aquarium fish sold in the United States today, and it's genetically engineered. But there's some really fun things coming on the market now. There's a pink pineapple that has been genetically engineered to be, instead of yellow, it's more the color kind of a cantaloupe. And it's really sweet. It doesn't have kind of that tartness of yellow pineapple, which is kind of a cool application. And of course, there's long-standing papayas that don't get disease. And I actually have out in my garden here some purple tomatoes that just came on the market this year that you can grow in your own garden, and they're little kind of cherry tomatoes that have extra helpings of anthocyanins, which make them healthier for you than conventional tomatoes. So there's a lot of different uses for the technology, and there's literally millions of farmers growing genetically engineered crops. And to date, we've had, I think, one food animal that's been approved globally after 35 years of having the technology in livestock, and that's the fast-growing salmon. So it's a bit of a disappointing story, I think, and one that I hope is changing as some of the new possibilities with genome editing come online.
Well, that's a really interesting point, Alison, because I know I can't remember who it was, but I remember someone recently talking about, the old food production issue, and that back in the 70s, there was multiple scientists saying, we're going to run out of food, the world's going to starve to death as our population increases and land gets used for other purposes other than agriculture. And we've now got a surplus of food in many parts of the world. So, yeah, I think they essentially attributed it, which I'm guessing that's what you're suggesting as well, to the fact that we've been able to improve the plant species and the survivability, and also, as you said, the footprint of that food source.
Yeah, although I would argue animal ag has a pretty good story to tell too. So we often hear, corn yields have gone up. And I'm like, well, you should have a look at what we've been doing in our animal species too, because I think we have an equally impressive story that perhaps isn't as well known as crop yields and Norman Borlaug, for example. But, a modern dairy cow is nothing if not miraculous in terms of her productivity per year. And of course, that reduces her footprint per glass of milk relative to what it was in the 50s. I think it's a third now of what it was back then, because she produces basically more than 4 times the milk she did in the 50s. And that's just very much genetics, but also, nutrition and veterinary care and what have you. But it's a super important story. And people don't think of genetics when they think of sustainability. They think of, carbon markets or whatever, but they don't think, I should probably call my geneticist to see what we can do to improve our efficiencies here.
Today's episode is brought to you by AgriCoach. You've worked bloody hard over the years and you've built a solid operation. But as your children grow up and start their own journeys, you're beginning to think about the next 5, 10, and even 15 years and how to keep driving the business forward and ensure a smooth and successful succession plan. Preparing the business, preparing the next generation, and preparing for your eventual retirement all requires a solid plan and a proven process. Unsure about the right steps to take? Head on over to agricoach.com.au/webinar to learn more about our Succession Proof Your Family webinar. AgriCoach, aligning families, growing wealth, and protecting legacies. And now, let's get back to the episode. Yeah, because biotechnology, correct me if I'm wrong in hearing this, is that it's a combination of of gene editing, or those sorts of technologies, shall I say, and correct me if I'm wrong here, Alison, because I'm way out of my depth, but the gene technology, but there's also breeding and measurement over many years. Is that correct?
Yeah, I mean, you could definitely put that under the definition of biotech because it's science getting applied to life. Traditionally, I think conventional selection, and by that I mean not using genome editing or genetic engineering, but just, EBVs and perhaps genomic testing to improve the accuracy of your EBVs, that traditionally hasn't been considered to be modern biotechnology. I guess it's old-fashioned biotechnology, although, if you look at our breeds, we've made tremendous changes in them relative to their ancestors, like dogs and the wolf, or auroch is the ancestor of cattle. So I would argue we've made more changes to them using conventional selection. If you think of a chicken today versus, 50 years ago, than we've actually made with any of these genetic engineering. Typically with genetic engineering or genome editing, we're working on a single trait, like, not growing horns or something. But if you look at an Angus versus an Allure versus a Drought Master. I mean, there's so many differences between them just based on conventional breeding. I would argue it's a more powerful tool that has been able to really alter our animals dramatically from their ancestral type.
And I'm guessing too that with AI coming through, processing power of computers, technology starting to rapidly change, that I'm guessing the gene editing or the genome editing side of things must be accelerating. Is that correct?
Well, I had to laugh because you used an acronym there that you assumed was a modern acronym. And when I hear AI, I'm like, oh, okay, well, artificial insemination. So how come they get to steal our acronym? We had that in the 1940s is when we first did AI in cattle. And now it's just assumed to be artificial intelligence. But yes, 100%, like artificial intelligence, I assume that's the AI you were talking about. Actually, there's papers on the AI of AI now. So using AI to do better AI insemination, that is. And so certainly all of those are coming together to both suggest which might be useful genes to edit. And so that's kind of more on a functional basis and say, well, let's, what happens if I knock this one out, or I alter this one, or I make it the same allele as what's in Jersey, or that type of thing. That's kind of the question at a fundamental level as a researcher that I ask. And then let's just say it does something amazing. Then it goes to commercialization, and that's typically in collaboration with a company of some sort who then has to figure out the trickier part, in my opinion, which is how do we disperse this technology to the broader industry? And of course, that very much depends on which animal ag industry you're talking about. So pigs and chickens are, super different to beef cattle, for example, in terms of the industry integration and breeding companies' role relative to producers making selection decisions themselves, which is much more beef cattle kind of idea behind their industry.
And I'm guessing also government regulation around these sorts of things, I would assume would be either difficult to deal with or a very slow-moving beast. Is that correct?
Just wanted to poke the bear and see if I bit back. So yes, I have spoken a lot on the editing regulations and more generally regulations and the importance of making sure that they're proportional to risk and not overly burdensome to where they basically stop the technology moving forward, which is what I would argue happened with genetic engineering in animals, where there was a multi-million-dollar regulatory package that it wiped out all the small companies and academia. It just left it the purview of large multinational corporations that everybody hates. And it's like we had an opportunity with genome editing to change that narrative. And actually Australia, to their credit, the genome technology regulator down there has said, if you're not introducing any novel DNA, we're not going to treat you any differently to conventional breeding, which totally makes sense because basically conventional breeders are operating on genomic alterations. It's just ones that nature introduced. And there's no novel hazard associated with genetic alterations per se. And so I think that there's hope there, and certainly the crop breeders have already started moving in Australia and bringing varieties to field testing that don't contain any novel DNA. They've maybe inactivated a gene that gives them a particular characteristic that they want in their crop, and we can do similar things in animals. And if we don't have an overly burdensome regulatory process, we're just still checking it's safe, as is true for all food, then I think it opens up much more opportunity for small companies, academia, to do some really innovative things that maybe a large company only wants to focus on, corn or, the big boys, versus, avocados or macadamia nuts or all of the other things that you might want to, introduce disease resistance to, for example.
Can you just explain genome editing, just so we're really clear for everyone listening, because not everyone's got a science background. So genome editing, what does that mean?
So genome editing refers to basically a set of technologies that allow you to introduce, in a very targeted way, a double-strand break in the DNA helix. So you probably remember back to your basic biology, and there's the C's and T's and A's and G's that make up a gene that produces a protein that does something. So if you can go in there and target a particular gene and do a double-stranded break, you have the opportunity to disable that gene, so knock it out is what it's called. And you can, for example, knock out a gene that is the receptor where a virus gains entry to an animal. For example, there's a pig where they've done this. They've knocked out the receptor, so that pig is no longer susceptible to porcine respiratory and reproductive syndrome virus. It's a virus, basically. So that landing pad is no longer in the pig. So the pig's fine otherwise, just living its piggy life, but it's resistant to that disease, which is not in Australia, but it's a very big, costly disease in the rest of the world. And so editing is using, they're called nucleases, which is just a fancy word for scissors that cut nucleic acid, and you tell them where to cut. And that's what the unique aspect is the precision with which you can tell it where to cut. Anywhere in the 3 billion base pairs that make up the bovine genome, you can go in there and say, go ahead and cut at this gene, because I know that's what makes my cows susceptible to bovine viral diarrhea or whatever I'm after. And I can target that gene and make an animal that's no longer susceptible to that virus. And that's the power, is that precision with which you can make that double-stranded break. And most people have heard of CRISPR-Cas9, or at least my tribe has. That's kind of the most famous of the editors. There's other ones that are around, but that one got the Nobel Prize, I think 2 years ago. And it's kind of the sexy one that everybody likes to use because it's relatively easy to use. Like even my lab can do it, and it's not very expensive to use. And so you can do a lot of experimental basic research with it without having to take out a big loan.
Now, when we had breakfast in Rockhampton last year, I know I asked you the question about what cool and exciting stuff you're working on, and I'm going to do that again for all the listeners. But one of the things that I noticed about you is you're really passionate about what you do. And when I asked you what are you excited about, I saw the energy levels jump. So what are you working on at the moment, Alison, that you're super excited about that's really cool, shall we say, in unscientific terms, or something around the world that people are working on that you think could have a massive impact?
Yeah, and so when you think about genome editing, there's some obvious single gene traits like disease resistance, for example, that come to mind, or making, for example, the northern beef cattle herd 100% dominant polled, using genome editing to introduce that allele or that variant that exists, for example, in Angus, and bring that into, Brahman, for example. So there's a lot of single gene traits which I think are really interesting and exciting, especially polled because it's dominant, and so you just need one copy to not grow horns. But I actually think the things that are kind of a little bit more whoa are things that can alter breeding program design. And so, particularly there are industries that don't want one sex. So, for example, layers and egg producers don't want boys, and dairy producers aren't all that excited about them either because they're not the productive sex. And so we basically produce 50% of the unwanted sex in those two industries. What if we could rather use either genome editing to sort the eggs so that the boys don't ever get incubated, so we never have male chicks hatching, or instead of sex sorting semen and paying quite a bit more for that straw and having the knockdown in fertility, what if the bull was only delivering Y sperm or X sperm, depending upon which way we wanted to skew? So for a terminal sire in the beef industry, we would prefer all boys if we're not getting replacement heifers coming back. And so messing around with editing to produce single gender or sex offspring, I think is really exciting, especially for some of the bigger cattle populations on Earth. And I'm thinking specifically here of India, where it's between the buffalo and the cattle there, that's the biggest dairy producer in the world and also the biggest bovid population. I think it's 300 and odd million. And in that country, of course, you can't eat meat or beef specifically if you're Hindu. And so the boys in the dairy industry there literally have no value. You know, at least in our dairy industry, we can produce, you know, we can maybe do a beef on dairy and get a young calf that we can go into the beef industry with. There, they really have no use whatsoever. And what if you could only produce females from that huge population? You've doubled the efficiency. Right? Just with one little switch. And so there's a couple of projects that I'm working on to try to produce all male offspring using, there's just one gene in the mammalian genome called SRY that's on the Y chromosome that makes you go down the male pathway. Typically, you get that Y from your dad. But we know in mice, and actually there's some human examples of this where there's been what's called a translocation, or that gene has got moved onto the X chromosome, that you can have XX individuals that have the SRY gene that are fully male to appearance. They're infertile because there's other genes that are required for fertility on the Y chromosome. But for a feeder animal that's going to the feedlot and off to the market, you don't need fertility in that case. So there's some interesting things there, and also things where genetic engineering, we're trying to incapacitate the sperm of one sex or the other so that the bull himself is shooting only Y chromosomes or only X chromosomes. So those are, I think, some really exciting applications.
How far is that technology off, do you guess?
So there has been reduction of concept in mice. Everything works in mice. It's just a rule of science. Everything works in mice and it never translates into large animals. And so we have like XX male mice, no problem. And there have been some genome editing studies where they've knocked out SRY in pigs, which, if you do the math, means that now XY pigs develop as females because they don't have SRY to take them down the male pathway. And that's a concern for the castration issue. So a lot of Europe will no longer allow pig castration because it's relatively invasive, if you know anything about pig testicles. And so if you could just have females, they grow as well as males in pig systems, that would be preferable from an animal welfare perspective. That's been reduced to practice in pigs in Germany. And then there are groups that are, I have seen a recent paper preprint that was on bioRxiv where they've got mice that are genetically engineered to basically inactivate one sex's sperm so that those mice will only produce one sex. And what's important is that it happens before fertilization, so you don't affect litter size. So you can't have it so half of the animals die, especially in a cow, because it has to happen before the fertilization. And so that's kind of exciting. And that paper just got put in bioRxiv a couple of weeks ago. So, you know, if it's happening in mice, it always takes a few years to get across to livestock. But I think the value proposition for that is so big that I really hope it's something that captures the imagination of researchers beyond the mouse community. There's a lot of stuff that gets done in mice that would be awesome in agriculture, but you don't necessarily have those people interested in translating it. And it's, you know, a mouse is, I don't know what it costs to keep one, 50 cents a day, versus large animal research is a lot more expensive. And so it gets difficult to actually get it translated across into large animals.
And when you've got large populations as well, like if you, as you said, in Northern Australia, et cetera, with the large beef herds, it can have a significant impact on production, couldn't it?
It could, especially if you could get it delivered by the bull. And that's one of our big difficulties with extensive pastoral industries like sheep and beef, is AI, as in artificial insemination, it can be used, and certainly in the upper echelons of the seed stock it does, but for commercial production, for the most part, in both California and Australia, it's being done by the bull. And so what can we do to equip that bull with better genetics?
And that's where we go into your excited topic. So I know you've been holding the horse back. Go for it. Talk about the most interesting one.
Yeah, well, it's one that kind of blows your mind a little bit as to how this works. but there's a concept called surrogate sire, or sometimes I hear it called surrogate germplasm, because it doesn't have to just be the male end of things. But the idea here is that a commercial bull out in the pasture with cattle is maybe tropically adapted and he's got influence and all the rest of that good stuff. But maybe what you would like for his calf crop is Angus F1. So you would like his genetics to be Angus, but you'd like his phenotype to be tropically adapted so he can survive in the north. So the idea is that you can use genome editing I'll use Brahman as an example of a tropically adapted animal, to inactivate the pathway that produces sperm in a Brahman bull. So great, I've now produced an infertile bull. Well done. That's really helping things. But, and this is what's important, if you then are able to introduce a cell line from an Angus bull, for example, that can go into the germline, or if you want, populate the scrotal sac of a Brahman bull, he can then be shooting Angus semen with his apparent Brahman phenotype, and all of the offspring would obviously be sired by that elite animal that you use to complement the germline of that bull. And that's amazing. So now I can do AI in the extensive northern pastoral situation, for example. But the other one that kind of builds on top of that, and this is where it gets a little bit like, okay, I'm not sure about all this. So we're basically beholden to the generation interval of a cow. And so bulls maybe mature at 12 to 18 months. Cows, similar ballpark. Maybe they've got their first calf and weaned at 2 years or whatever, something like that. That is the soonest we can get to the eggs of that cow and the sperm of that bull. Maybe you can make them a bit quicker if you give them hormones and stuff. But we're waiting for basically adulthood to hit the cows and bulls. What if instead we could take cells in a petri dish and basically use genomic selection to pick the very best animal in terms of the cell line of a male and a female. So this is a really good cell line of both. Now I'm going to expose the male line to undergo spermatogenesis in a petri dish, and the same for the female line. So now I've got an egg and a sperm in a petri dish. I'm going to fertilize them. I'm going to produce a new embryo. I'm going to test genetically from a genomic selection. Is this better? Hopefully it is. They were both really good parents. You know, who got the best selection of Mendelian genetics? Same way we do in an animal that's alive. And then you can say, okay, well, you're the best bull in this lot and you're the best cow. Let's do it again. And you can, if you do the math on what's conceptually possible there, you can get 3 generation intervals a year instead of a generation interval every 2.5 to 3 years. So you've just basically started to time travel to visit your great-great-great progeny in a time frame that's still within your lifetime. And it really opens up some interesting ideas around the seed stock sector and what that might look like. And am I just like really out there? Has this ever happened? Yes. It's been reduced to practice in, guess which species? Ben, come on.
Mice.
Mice. It works in mice. Therefore, it's going to work in cows. So it's a long way from commercial practice at this stage, and in fact, even reduction to practice in cattle. So just to be fair, I don't want everyone's expectations to get raised. However, if you can reduce the generation interval, that's one of the main drivers of the rate of genetic improvement. And it's inversely proportional to that, meaning that the shorter the generation interval, the more rapid your rate of genetic gain. And so you can turn basically a cow almost into a mouse in terms of its generation interval using this technology. And I think that's a super interesting application. And it doesn't even require necessarily genetic engineering per se or genome editing. Like you could just do all of this just using conventional selection, conventional in a petri dish, but yeah, you're not using any modern molecular techniques. And so you don't even necessarily have to go down the GMO pathway to make all of this work. And I think some of those applications are really cool. And they're doing this in chickens and fish and pigs and cattle. And the fish people, and fish are really cool because they really easily take up cells to be the new germline, the surrogate sires, and they're much easier than mammals. And there's some really interesting applications in aquaculture where they're taking up genome-edited lines that are disease-resistant, for example, and then also infertile, so that the commercial fish are disease-resistant but can't spread their genes to the wild salmon populations, for example. So it kind of addresses some of the concerns around genome editing in terms of getting into wild populations, whilst also allowing the useful introduction of these different traits into these commercial sires in aquaculture. So lots of really cool stuff on the horizon there.
Because I'd have to think the disease management would be a fairly big one for, and I'm thinking about the foot and mouth scare we had a couple of years, or hopefully a couple of years ago. Is that sort of something that's really heavily being researched? Is how do we switch off a propensity to suffer from that disease within the animals?
Yeah, so if you look at the global genome editing literature as it relates to food animals, the number one target is production traits, yield. That's not that surprising, right? And especially because there's this one particular gene that's easy to knock out and you see this really dramatic phenotype, and that's myostatin, or double muscling, that creates the Belgian blue cattle. Not a great trait in terms of just calving problems, and it's got some other issues, but that's a really easy one for a researcher to go, look at my paper, here's a really muscly X, whatever X might be. But then there's all the reproductive stuff I just discussed is the second biggest thing. And then third is disease resistance. And to me, that's the traits that I think we as an academic community should really focus on, because I think it ticks the three boxes of sustainability in that it's better for the animal and the farmer by definition, better for the environment, so no dead animals, and hopefully more profitable because no sick animals. And so it's one of those rare triple wins, and it kind of puts you in a situation of, if you don't want that, then you're kind of arguing for disease susceptible animals, which is always, I always like for there to be an awkward trade-off that people have to really think about. Well, what is it that I value most? Is it actually, never, ever, ever allowing technology to come into the market, or is it actually addressing problems that are causing illness in animals, or what is it that I really value? And I think those kind of implicit trade-offs of some of the sustainability metrics are really evident in some of these applications, and that leads to good discussions, because it's not just a no-brainer of, everybody agrees this is a great way to do this and we should just do it. There's always going to be this kind of conflict and tension between the social aspects and the economic aspects and the environmental aspects. And so I think ones that make you think hard and scratch your head and go, ooh, I'm not sure how I feel about this, is actually a really good example, because it makes you really kind of nut down on what your actual values are. What do you value the most here? The
obvious thing that most people talk about, Alison, as you'd obviously know, is when we talk about these technologies, is the fear of the unknown. What are the risks? Are researchers really focused on the risks? Is that a really high priority? Or is that pressure usually coming from outside government regulators, etc.? Well,
I would argue that we being academics do quite a lot of the risk evaluation. A little bit, we do what we can get money to do. I'd like to pretend like there's academic freedom, but at the end of the day, I have to find a call for proposals that suits my interests, that I can make a convincing case to that funder to give me money, right? That's kind of how it works. And there are some programs here and certainly in Europe as well, and I'm not familiar with the landscape too much in Australia to be able to comment, that are the Biotech Risk Assessment Program that gives you funding to basically do risk assessment work on your experiments that you're doing. And so, for example, we did a project. We were collaborating with a company called Recombinetics to introduce the polled gene, or the not growing horn gene, into dairy cattle background. And part of that was also a risk assessment grant where we did whole genome sequence on the animals to see if there was off targets, or, if there's edits had happened anywhere else. Did that allele introgress somewhere we didn't want it to? We looked at the health of those animals. We phenotyped them and they kept not growing horns. It wasn't a very interesting experiment, but we also looked at the milk and meat composition. Did it affect that at all? And not that you would necessarily think it would, but we have data to show it doesn't. And so now hopefully nobody else has to do that experiment, because we have the data to show it just affected horns. It didn't affect milk and meat composition, for example, and the animals were healthy and fertile, and all the other traits we care about were unchanged other than not growing horns. So we do that. And I would argue, if you've got a project that's been funded I don't probably care where you are in the world. It's gone through multiple levels of scrutiny, be it not only the funder likes the idea, but it's gone through your animal ethics committee for you to even do the research. There's numerous kind of checks and balances to make sure that the research is kind of in line with generally recognized ethics, I guess, for want of a better expression. So I feel like for the most part, academic researchers are pretty serious about this type of stuff and try to get the data to answer the questions. And they're not just cowboys out there going, oh, I should be right. We're actually providing the data and doing sometimes pretty silly experiments to get that data, like, meat composition of a polled animal, whilst you're eating an Angus burger for lunch. It's like, what are we expecting here, guys? So, yeah, that's what we do.
But again, I think one of the best things for the industry is for people to understand, like the transparency, understanding that it isn't just a bunch of cowboys essentially just saying, wow, that sounds like a great experiment. Let's do it and let's roll it out. There is multiple layers of not regulation, but ethical checks and balances.
That right there is just basic research. But if you wanted to bring it to market, there's also multiple layers of regulation as well. So there's more to it if you want to commercialize something. And to be honest, introducing edits into commercial populations, it's no mean feat. So Genus, which is a breeding company that works in both cattle, you're probably familiar with ABS, the American Breeders Service. They also have the PIC, the Pig Improvement Company. They have outlined their intentions to introduce this disease resistance allele for the PRRS, the porcine reproductive respiratory syndrome virus, into their populations. And if you don't know anything about pig breeding, it's a 4-way cross. So there's 4 grandparent lines and 2 maternals. They come together and then they cross again into the 4-way full-on heterosis, commercial pig, right? That's how you're meant to do these things. And so they needed to introduce that edit in a homozygous, as in both alleles edited, so mum and dad's, in all 4 of those grandparent lines in multiple animals in order to breed it down into their commercial setting. So we're talking hundreds of animals that they needed to edit and fully characterize. And that's one breeding company with one species for one line of disease-resistant pigs. And so if you think about it, just think, Northern Australian cattle population, there's multiple breeds, there's a lot that would, if you think about how you would introduce it up there, you're probably going to have to go some sort of AI or surrogate sire, and it's going to have to be in different breeds. And so that end of it is kind of the fascinating and the translational aspect of science. But it's also really important because it looks great on paper in, my peer-reviewed manuscript, but if it can never be used in industry, who cares what you can do in a mouse, really? And that I think that translational aspect is what interests me, and it has to be a very valuable trait to the industry to make it pencil. And obviously the pig people think that PRRS resistance is going to pencil, or they wouldn't be doing it. They're a publicly traded company, so they're going to be beholden to the shareholders, and they must have done their math and figured that out.
It's amazing. So Alison, if you had control of all the money and you could do whatever you wanted and study whatever project, where would you deploy that?
So I'm looking for low barriers to entry. So I would go to countries that have sensible regulations. And that's a very pejorative term, which probably suggests I'm not going to stay where I'm at right now, because I don't think the US approach is fit for purpose for regulating of genome editing in animals. But the countries that have got it right, in my opinion, are mostly the South American countries, and specifically Brazil and Argentina. And if I was really going to roll this out at scale, I think I would probably be a younger person. I would just move my lab to Brazil, partly because they have the biggest cattle population on Earth. If you don't count the buffaloes, they knock India out. But they also are just absolute techno junkies, and their rate of AI, artificial insemination I'm talking here, in their beef cattle industry is higher than their dairy cattle industry, which is like, what? And they have these huge synchronization protocols. You've probably seen, like, they've got so many people out doing assisted reproductive technologies in their beef cattle population. And they've got the expertise to do all the embryo transfer and what have you. And it's like, that's just a perfect storm right there. And that's an industry that their cattle are not as efficient, or they don't yield as much as some of the more developed countries do, and obviously huge exporter and under the gun for all sorts of greenhouse gas infringements. It's kind of oh my gosh, if you could make change there, you could really make a difference. And so that if I had all the money in the world, that's probably what I would focus on. And I wouldn't sneeze at Australia either. I mean, I think you guys have a sensible regulatory system and, obviously some big pastoral companies that could utilize some of these technologies to their benefit. And if you could just wave a magic wand and make everybody polled, why wouldn't you? I forget what the death loss is, 3 or 4% to dehorning in northern cattle. Just eliminate that, and it's one of those win-win situations. But I also acknowledge it's not that easy. But yeah.
It's interesting, Alison. I heard something the other day, someone talking about drones and how the regulation in America was so tight on drone technology. It was completely locked down. But in China, you could do whatever the hell you wanted within reason, and as long as it aligned with the CCP. And then what's happened now is the drone market in the world is dominated by, I think it's DJI, which is a Chinese-owned company. And so we've got to find that balance between protecting and being careful with whatever technology we produce, but we also don't want to be so locked down that we lose our competitive advantage. And that's something I'd say to Australia is, we have so many competitive advantages in the fact that we're based in a different part of the world. So we've got that bio risk is lower than everywhere else. We've got better standards. We use better animal breeding technology, but that could be eroded fairly quickly if other countries decide to embrace such technology. So I think, it's something that Australia should really be considering. And 9 times out of 10 comes out to finding the right people and the right amount of money to be able to do the research.
Yeah, and I think especially given the pace of innovation in Brazil, I would suggest is probably one of your major competitors. I don't know if that's actually true in the export market. Certainly they're a big exporter. I'm not sure if they do the quality of beef that necessarily Australia might get the premium for. But I look at that and I think, here's a technology, genome editing, that was developed at Berkeley, an hour down the road here by a public sector researcher. And the benefit is potentially going to go at least in agriculture, to countries other than this one. And it doesn't make sense for a publicly developed technology to not be utilized in the countries that paid for its development. So it's very frustrating. So I think such an important aspect of governance to make sure regulations are risk proportionate and fit for purpose.
Yep, and still allowing innovation and not quashing innovation, because I think that's one thing Australia's got is a very innovative bunch of producers out here. And I know I've interviewed my mate Jeff Burchell a couple of times around using all different reproductive technologies, and there's just this drive in the next generation, Alison, that I'm sure you're seeing is to embrace different technologies, different measurement, genomics, those sorts of things. And I think we need to embrace that. So, Alison, I'm very conscious of your time. You're a very busy lady, and this has been a whole lot of fun. But where can people find more about you online? And also, I just want to touch on, you're going to be at Beef this year, Beef 24 in Rockhampton. So where can people find about you online and talk me through Beef this year? Sure.
So online, I'm at my university website. So you just have to remember how to spell Van Eenennaam. So you'll be fine with that. No worries.
I'll put that in the show notes.
Yeah, probably Alice and UC Davis will get you there. But kind of more social media wise, I'm on, well, I still call it Twitter because I'm old-fashioned, but X as BioBeef, BIOBEEF. And my content is almost all about genome editing, and I don't stray from my designated path there. And then in terms of Beef, so super looking forward to coming up to Beef and going to be participating in 2 events there. So the Beef Australia Monday morning breakfast, I'm going to be speaking at that. And that the overarching title of that is on being influential. So we're here from a range of different players there, from different countries and different industries in terms of trying to get your message out, I think, in general. And then on Wednesday afternoon, I believe at noon to 1:30, I am going to be doing a session called Outnumbered, which is basically a fun look at EBVs and how they've been implemented in basically all of the animal protein industries, and particularly our competitor protein industries, chickens and pigs, and their incredible value at selecting animals that are the best to be parents of the next generation, which basically is our job. And you saw me speak at Rocky. It's not there will not be a lot of equations on my slides, but there will be a lot of interesting pictures. I'll just leave it at that.
I think we got to meet Elle McPherson last time, I think it was, from memory.
She might be making a bit of an appearance. I think you might also recognise some producers are going to be making some appearances here, and actually a number of dogs. And I'll just leave it at that.
Fantastic. Well, I can tell you, after listening to Alison last year at the Frontier Genetics launch, I can tell you what, it's incredibly entertaining and also incredibly informative. The stats that you were running through when you did do the stats were mind-blowing, and the story you were able to tell around that was exceptional. And that's why I wanted to get you on the podcast today, because I think there's very few scientists that you come across that have the ability to communicate as well as you do, Alison. I think you're doing great work, but I also think it's fantastic to see how that you're able to explain these things in very simplified terms so everyone can understand, and also have the ability and have the desire and the want to talk about not only the opportunities but also the risks and the way we should be looking at it as an industry. So before we sign off, Alison, I've got one more question that I like to ask all my guests, and you probably haven't had a chance to prepare for this, so it's going to be interesting to see what pops in. But if you could go back in a time machine, and instead of going forward like we talked about before, but you can go back, say, 20 years in time, what's the one piece of advice you'd give a younger Alison?
20 years?
Well, let's go 30. I don't mind.
We might want to go 40, I think. So
go 40.
Well, actually, maybe 20 years is a good one. I think just because maybe my story is a little bit atypical in terms of scientist, and so I'm 60, so we can do the math. So that I would have been 40 20 years ago. And I was only 1 year into my faculty position. It basically took me 39 years to get a job. And, if you told someone that you weren't going to start your job or as a faculty member until you're 39, that would sound like that's crazy. Like, you'll never be successful in that path. But that's just what it took for me to, do all my education. I had jobs doing just everything. I was a wool classer at the wool board. I was a barmaid. I was an extension agent for a number of years. I was a scientist at a company here. Just took me a long time to find the right job for me. So it was 39 I was when I started that job. And I remember the first grant application I put in, it was actually genetically engineering cows to make high omega-3 milk. So for a health claim on the milk. And the reviewer said that it was technically a very good proposal, but that the public would never accept genetic engineering, and so they weren't going to fund the proposal. And I just remember curling up in the fetal position thinking, I'm never going to be successful at this job because, everything's against this idea. And actually that pushback kind of opened up the whole science communication end of things and made an opportunity that I didn't see when I was curled up in the fetal position thinking I was never going to be successful. So you're never too late to start a new career, and just because you kind of meandered around a bit and traveled the world and did some unconventional things doesn't mean you've destroyed all possibility of having a successful career. So I guess I would tell myself that.
Yeah, I think that's some great advice, especially for a lot of people out there, because a lot of young people especially can find themselves a little bit lost. They don't know what they want to do. And I think that's a great story. And mine's very similar. I spent 20 years, Alison, as a financial advisor, ran my own business. And then in my early 40s, I'd made a change to podcasting and coaching. So
there you go.
Age is no barrier. There's plenty of opportunity out there. You've just got to find what you are passionate about and what you're good at as well. So that's some great advice, I think.
Yeah.
Excellent. Well, look, we're done for today. I've got, again, as I think I say every episode, I've got a page full of notes there. I'm really looking forward to beef. I think as we've recorded this, we've probably got just a little over a month. I'm hoping to get this episode out before beef. Yeah, and I'll look forward to seeing you there. Is there anything finally you'd like to leave for the audience before we head off, Alison?
No, I mean, other than I hope I've given Australia's OTGR, the Office of the Gene Technology Regulator, the thumbs up. I think there's opportunities to use genome editing in animal agriculture in Australia, and I know there's a little bit of interest from different groups, and I really hope that takes off, and because I do think there's some really interesting applications that could be beneficial for the industry.
Fantastic, Alison. Well, look, thanks very much for your time, and I'll see you at beef.
All righty. Thanks a lot, Ben.
Well, I don't know about you. That's a bloody lot of information to digest. I'm probably going to go back and have a listen to that episode because there was a whole lot of information, and I think listening to Alison, you can see how she's really passionate about the industry, about the opportunities for us in agriculture, but I think also more importantly, the animal side of production. It's also really clear to me that Alison's very clearly understand the risks and the ethical considerations, and there's a whole lot of regulation and industry, I suppose you'd say, guidelines to make sure that that is paramount in all of this research. What I would want to share with the industry is these are my thoughts. I think our competitors are going to be embracing these sorts of technologies. There's going to be pros, there's going to be cons, there's going to be risks, there's going to be opportunities. With any new technology, that will always be the case. But I think if our competitors, like places, as she mentioned, Brazil, are going to be embracing this technology, we would be crazy not to be at least investing some money, time, and research to be able to get this right. I just want to say a big thanks to Alison publicly. She's a very busy lady. She's very much in demand, and she graciously agreed to come on the podcast before she comes over for beef. If you are going to beef, go check out her talks. I can absolutely concur that not only is it informative, but she does it in such a entertaining way. She got a lot of laughter at the Frontier Genetics event I did last year. So do yourself a favor, go along and watch Alison. She's a really great researcher and entertainer. So if you think anyone in the industry that you might happen to know might be interested in this podcast, we'd absolutely love it if you could share it with them. That's the way we share the wealth and wisdom. If you've got anything that you'd like us to do a podcast on, any topics, drop me an email, ben@thefinancialbloke.com.au. But above all, jump onto Spotify, jump onto Apple Podcasts and give us a 5-star rating. That helps us spread the word and get the wealth and wisdom out to more people. Well, that's it for today. I hope you've enjoyed today as much as I have. We'll see you for the next one.